right, continuing on with the TMCC Library Open Genealogy Lab Outstanding Guest Speaker Series, today we are pleased to welcome back Marleen Van Horne. Marleen retired as a systems analyst from the telephone company and has devoted several decades of her life in pursuit of her family history. She is an expert at using Family Search and administers 16 different surname projects at Family Tree DNA. Today, Marleen is going to explain the recent Y-DNA haplogroup R-L754, also known as R1b1, split that was recently announced and how it will impact our genealogical research. So I'd like to offer a warm virtual welcome to Marleen.
Okay. Suzanne asked me about talking about the, I'll get this out of here too. Well, okay. Talking about this split that has taken place in R1b1 and the new RL, I think it's M754. And so what I'm going to talk about before we ever get to that is I'm going to explain about single tandem repeats, which are short tandem repeats, which are the markers that determine matches in Y-DNA, and single nucleotide polymorphisms, which are the SNPs that have caused this split in the R1b1 haplogroup. So that's where we are. When you do a Y-DNA test, and first let me say that as far as I know, the only company offering Y-DNA tests is Family Tree DNA. And this is what it looks like at Family Tree DNA when you're Y-DNA tested. Over here, I've cut this off because over here on the left side is where the individual's name is and their kit number and who their ancestor is. But I did leave the haplogroup on, and this is the header for the tables that show your DNA matches. And it happens that these three men, one of these men, it was my half brother, and the other two men are second cousins to my half brother and I. So this is my family that you're looking at. Not that it makes much difference. Okay, at the top of the chart, when you look at this table, you'll see all of these names across here. And each one of these names identifies a location on the Y chromosome. Well, let's go there. Okay, so here we have the chart again. Now, if you think of these names as like off-ramps on Highway 80, you know, as you're driving across the country, you come up on these off-ramps. And just before each off-ramp, there will be a bunch of signs telling you how many services are available at that off-ramp. If you think of the Y chromosome in the same way as the marker identifications being like identifications of off-ramps, and then the numbers displayed under each marker as being the number of services available at that particular location on the freeway, you'll get a good idea of what the Y chromosome is like. And it happens that when you do a Y-DNA test, they use a PCR machine to analyze each person's individual DNA. And these locations are previously identified. You can't exactly just go in and look at Y-DNA and come up with something. So these particular locations listed across the top here have been deliberately selected. And what they're looking for is the number of times the nucleotides C, G, T, and A, and I never can remember what their full name is, so I always repeat the letters. And most people just talk about them as letters. What the nucleotides C, G, T, and A how many times those 4 nucleotides are repeated at that particular physical location on the Y chromosome. So at this first one, which is identified as DYS 393, those nucleotides repeat themselves 13 times. And these, each one of these locations is called a short tandem repeat. Now I put an example down here for DYS 455, and I used it because it was short. It only has 8 repeats. And so down here below, if you were looking at the raw data in the location where DYS 455 appears, you would see this 8 times. And these numbers up here are known as the repeat values. In other words, they tell you how many times the 4 nucleotides repeated themselves at that physical location on the Y chromosome. Okay, so then well, there we go. All right, here, whoops. There, we've got the example of the test results up here still. Now, these particular markers, there's something like, I don't know, there's a lot on the Y chromosome. Sometimes I've heard 800, sometimes I've heard more. And when I tried to look up specifically how many markers there were on the Y chromosome yesterday, I couldn't find it. But these markers were specifically chosen. The ones over here on the left that have the dark blue and sort of the muddy red background on the names, these markers are the oldest ones on the Y chromosome. And initially when Y DNA testing was started, it was thought that that was all you had to test and you'd know everything you needed to know about the Y chromosome. Well, they very quickly found out that that wasn't right. These are too old. And these are markers 1 through 12. You know, when you go and you look at, and they ask you what kind of a test you want to take. And I think one of the choices is 1 through 12. Well, don't waste your money on that one because the information is too old. If you match somebody on markers 1 through 12, like right here, see all of these guys match. If you match somebody there, you're allowed 1 mutation and you're still considered a match. Okay, then and those markers come from about 40,000 years ago or older on the Y chromosome. And that's why a 12 marker test is really not, it's not informative. And if you have been Y DNA tested and you're looking for your surname and you get down to 12 markers and that's the only place you find your surname, well, don't bother to waste your time there because basically it means the common ancestor you had with that other fella lived 40,000 years ago, probably. Okay, then the next group starts here. No, it starts here and it goes up to here. And these markers are chosen from the markers that were established on the Y chromosome between 25,000 and 40,000 years ago. And so that's starting with marker 1 here and going through 25 over here. And you're allowed 2 mutations in overall, and you can still be considered related. I want to point out this DYS 464. It can have up to 8 values. In this particular example, it only has 4. But if you go looking through the test results on family tree DNA, you will find in some instances where 8 numbers appear here. This is a a rather interesting group of markers, because after Y-DNA testing was started, it was discovered that these markers live on the Y chromosome in the area of a fertility marker. And if there is a mutation that for some reason wipes out these values and causes them all to be null, the chances are that that fertility marker has been damaged and the man is infertile. So for that reason, you're never going to see a set of repeat values for the DYS 464 marker that is 0. That is like through inspection, it's weeded out, and the individual who did that test is advised privately. Okay, so that gets us through 25. Then we come to markers through 37, and that gets us up to here. When I first started doing Y-DNA testing in 2005, 37 markers was all that you could do. And over the course from 1 to 37 here, you are allowed 4 mutations. And obviously this is like from 10,000 to 40,000 years old, because you're going backwards. These are the more recent mutations, and those are obviously the oldest ones. Well, sometime after we were doing 37 markers, they introduced 67, and we did 67 for many years until they ceased offering 67 markers, and they reduced the price on 111 markers, which had been running for a while, to the same price as 67. But there are still people out there that have 67 markers listed, and you need to know that over the first 67 markers, which brings us up to here, you're allowed 7 mutations, and that is about 10,000 to 40,000 years ago. You can find viable matches in here, and you can prove common ancestors very well with this group if you test 67 markers, which you can't do anymore. Now, the last choice that's available today is 111 markers, and I couldn't get the whole table on my screen. You wouldn't have been able to even see any of the repeat values down here if I had put the whole table on. So 111 markers starts here and it goes off the page. If you test 111 markers, these last ones, these light blue ones, are the most recent ones, and that'll bring your matches down to the present day. So when you match somebody from 1 to 111, and you match them either an exact match or a 1, 2, 3, maybe 4 mutations, something like that, you can be pretty sure that you are related to that person within the last 500 years. Now, the reason I say the last 500 years is this was designed so that if you match someone, you would have the chance of finding paper documentation to support the match. They consider genealogical time to be the last 500 years. And as time goes by, it'll get to 600 and so on, because that's the period of time for which we've had written records, or at least a lot of written records. And you have a chance of going back and finding supporting documentation for your matches. And each one of these is designed so that it is theoretically designed so that you can find somebody in the last 500 years. Now, on this chart up here, you'll see there's sort of pinkish red highlights and blue highlights. These are mutations that separate the three individuals here from one another. And these three, well, all three people have done 67 marker test. And among the three people, there's only 6 mutations, which means that they're related to one another. And since I know actually how they are related, it's sort of a moot point. This last one and the first one, between them, they only have 6 mutations. So they still come under the 7 mutation limit. Either one of these compared to the middle one, there's only 3 mutations involved. So there's no problem with that. It's assured that they're related. Now, these 3 men all have the same great grandfather. And they're from my Van Horne project, since they're related to me. And basically, these men have more mutations than anybody else in the project. Our great grandfather was in the Civil War, and he was at the Battle of Vicksburg, and he had typhoid fever, and he was in the hospital. He did not fight because he was sick in the hospital, and he had not married before he went to the Civil War. So all of his children were born after he married. And since the Y DNA in most of the Van Horns, which include all my other cousins outside of this example, is much more stable than the DNA of these 3 individuals, I make the assumption that his Y DNA was damaged as a result of the typhoid fever. And so if I were to put these gentlemen in the same grouping as all the rest of my cousins who descend from the common ancestor Mathis Corneliuson, you would see that these 3 have the most mutations of anybody. So the way you tell matches is you get 2 individuals, you set their test results on top of one another, and you look down the columns and you see if the numbers in the columns are the same or if there are any differences. And a difference like this indicates a mutation. When I choose to look at the Y-DNA chart, I always choose the colorized chart, because the colorized chart has the mutations highlighted, and they highlight them so that if the mutation comes from a red marker, it's kind of pink or red, and if it comes from a blue marker, it's blue.
Marleen, there's a question in the chat box that says, can you explain the steps on how you got this visualization off of Family Tree DNA?
Yes, I went to Family Tree DNA and I left-clicked on my mouse, and a box comes up that says screen capture. And it has you draw a box around the information that you want to capture. And then a little thing comes up and says copy, and you copy your box. And then I'm using PowerPoint here. I just went to the PowerPoint program and I pasted it into the slide for PowerPoint. Does that help?
Barbara, did that answer your question?
I'm still not sure how you got to see it. I mean, I can understand how to copy it once it comes up on FTDNA, but how did, I just don't understand the steps.
Okay. Have you tested at FTDNA?
My great uncle, yes.
All right. I'm not going to explain it here, but
Okay.
Make a note.
Maybe after class.
Well, make a note of my email address, and you can set up an appointment with me and I'll do an analysis for you and explain it all to you.
Wonderful. Thank you,
Okay,
Marleen.
because you're missing out on something. You don't know how to find the table that has this information in it. And so I'll explain that. I might, if you guys want, I'll show you how to find it after we're done with the recording. Okay, so here we have, we understand the short tandem repeats is what these are called. And these numbers indicate how many times the nucleotides were repeated at each location on the Y chromosome. And there's many more locations on the Y chromosome that you could test for. And these are the ones that have been chosen for the basic tests that family tree DNA does. Okay, now. Originally, when DNA testing began, the scientific community used the type of scientific naming convention that they had been using up to that time. And if any of you are a scientist, and I haven't got this right, just be gentle with me. That naming convention looked like this down here, R1, B1, A1, B1, and each letter was added when some type of an observable physical difference was noted in a living organism. I'm not a scientist, but as I understand it, this is the way everything was identified in anywhere, in any genome, plants, animals, human beings, I don't know, rocks, whatever. They use this type of a naming convention. First, they found major differences between individuals, and they put those major differences in groups. And that's what the haplogroup is. If you think of the phylogenetic tree, you'll know that it has all these branches off of it that go from A to Z. And those are the basic branches off of the phylogenetic tree that differentiate from one haplogroup to another. Then, having identified a group of individuals which were arbitrarily designated the R haplogroup, they then started looking for physically observable differences between them that would allow them to say, well, this one is different from that one, so we'll put this one in R1 and we'll put that one in R2, or whatever. And so when we first started doing DNA testing for genealogical purposes, we got these haplogroups that could stretch out to 16 numbers and letters like this. And over the first 10 years, from 2000 to 2010, it became noticeable that there was something else going on. And so this is the way it was originally done when we stumbled on genetic genealogy. Then, as I said, they noticed that something else was going on. And about 2000, the year is 2008, they realized that what was going on was another type of mutation, and that those mutations were a sort of random out of sequence appearance of single nucleotides. And this is called single nucleotide polymorphism. It's abbreviated as SNP, and it's pronounced SNPs. So when you start hearing people talking about SNPs, you'll notice that they're talking about these, quote, weird things that were happening. Well, it turns out that SNPs are almost more important than the short tandem repeats, but you need both of them. And the short tandem repeats are randomly populated by single occurrences of a nucleotide that's not in the pattern of CGTA. And let's see, so we'll go down here one more. If you think about the highway again, and the Y-DNA test results are the number of times the 4 nucleotides are repeated at a specific location on the Y chromosome. Those are the off-ramps for the highway. It was soon realized that these individual nucleotides are scattered throughout the Y chromosome, and they're like footprints down the Y chromosome, starting whichever way you want to start it, starting from today and going back to the beginning of human beings, or starting at the beginning of human beings and coming down to today. There are all these SNPs scattered all around, and they can't be exactly identified by the PCR machine, which is what's used to analyze Y-DNA or all DNA. And the reason they can't be exactly identified is that in the case of the PCR machine, you have to tell it what to look for. And that's why we have those locations on the Y chromosome, and it goes directly to that physical area on the Y chromosome, and it looks for how many repeat values are there. Well, when you have some random nucleotide just sitting there somewhere, the PCR machine cannot find it unless you tell it where to look. So over the 8 to 10 years, first 8 to 10 years that we were doing genetic genealogy, these SNPs started popping up. And the more people looked at them, the more they realized that these are pretty significant pieces of information. And they now know that almost everything important about DNA, as far as the information that it applies to living organisms, is contained in the SNPs. And the SNPs are searched for. It's sort of like fishing. The genetic laboratories that are set up to do this, they go out and they look at DNA samples, and the larger the sample is, the better. So you're more likely to find SNPs by doing a big Y700, or by going to some place who does the full genome test. You're going to find SNPs there. You're less likely to find SNPs in the up to 111 markers that are regularly tested to determine relationship. And in this example down here, if you had if you were looking for a SNP, you'd be looking for something that looked like this, where there's an extra A here that shouldn't be. It doesn't have to be an A. It can be any of the nucleotides. And these mutations actually form a timeline from the beginning of a human evolution to the present day. These mutations also are now being related to medical issues and other things. Anything that you find out about the human genome is probably related to information contained in a SNP. Now, there's SNPs in all DNA, Y-DNA, autosomal DNA, mitochondrial DNA. Those of you who have done a autosomal DNA test at Family Tree DNA will perhaps notice that it tells you the autosomal DNA will tell you how much DNA you share with somebody else, autosomal, And then it'll tell you how many SNPs are included in that match. And the PCR machine has to be told where to look for these. And as I said, the labs themselves do research on long genome tests and find the SNPs that are in there, find the SNPs, identify them, and they work up what are called primers. When you want to do SNP testing on DNA, you have to have the primer, and you put the primer in the PCR machine, and if that SNP is there, it'll find it. So when that happened, then something different took place. They stopped using like the R1, B1, all those letters and alphabets, and they started using the identification markers for individual SNPs. Now, my three cousins are all ISK1234. They all pass through that SNP. And I say pass through because there's more to it beyond that, but they are all that particular SNP. And that's what the SNP identifiers look like today. We no longer in the phylogenetic tree like that you'll see on family tree DNA. If you click on the haplogroup marker or the haplogroup name in the column, or actually it's on the homepage of a man's Y-DNA, you know, on their kit where the homepage is, and it tells you that whether they did Y-DNA, autosomal DNA, mitochondrial DNA, whatever tests they've done. Over on the right-hand side is a box, and it has a number in it, and the number is the same number that shows up in the table. And if you click on that number, it'll take you to the phylogenetic tree, and it'll show you where that number is in relation to the phylogenetic tree. Now, this is just an aside here, but almost everybody, for example, if we go, let's go back up here to just a minute. Let's go back up here to the table. Okay, see over here in this first column where it says haplogroup, it says IM253 and it's in red. That is only a hint at what the actual SNP for my ancestor, for my cousins is. Now, I happen to know that it's ISK1234 because I had it independently tested. And I didn't have it tested here at Family Tree DNA because I was too cheap to spend for a big Y. And I know, and I've mentioned before, that there is another company called YSeq that can test individual SNPs. And you have to know what to tell them. And they'll do a SNP test for like $20 as opposed to $400 or $500 for a big Y. But you have to know what to tell them. Well, we had enough of people in the Van Horne project who are cousins to me and these three gentlemen. And we had seen, that's where we first saw SK1234. So I went to YSeq and I asked Thomas Cron to test my brother for that specific SNP, and he turned up positive. So we know for sure that he's positive. He just wasn't tested at Family Tree DNA, so they won't put that SNP in here. But when you see it in red like this, this is pretty close to the phylogenetic tree trunk. This is not a true haplogroup. It's a suggested haplogroup. It's a predicted haplogroup, but it's not the actual haplogroup that your male relative belongs to. And only when you see a number in the haplogroup column on one of these results charts do you know if it's a real one. It will be in green. Now, sometimes they're in green and they're little short ones like this. Well, what that means is that when that individual was tested, they couldn't tell what the haplogroup was. So they went in and they did some special testing and identified the haplogroup, but it still might say IM253, only it'll be in green. And that just means that this suggested haplogroup was actually tested. Only when you see it in green and it's longer, like ISK1234, do you know that that's an actual tested haplogroup result? And it makes a difference because where this particular haplogroup ID rests on the phylogenetic tree is like maybe 10 SNPs below the tree trunk, where the actual ISK1234 is like 100 SNPs or 150 SNPs down from the tree trunk. So it shows it's way far away from the trunk. It happens that the I haplogroup is a very recent haplogroup, maybe 2,000 to 4,000 years old. And so when they started out doing haplogroup IDs and they started with the oldest ones, it took 3 tries before they ever got down to my brother and cousins, because the haplogroup that my family comes from is so recent. Now, I may have gone off the track here, and
Marleen, can I have you put it in presentation mode, because we're seeing your thumbnails on the side?
yeah, I know, I'll put it back.
Okay.
I couldn't get it to go up. There we are. Thank you. Okay. So let's go down here. So we're down here to the SNPs, and you all understand that a SNP is the insertion of a nucleotide someplace where it doesn't belong in what would be the normal repeat values. And then we get down here. So Suzanne asked me about this and asked me to do this because in the last few weeks, they announced that the haplogroup R1B1, which is very, very old, it goes back close to that 10 or 12, or the first 12 markers when you test it. You should find R1B1. Well, it was announced in the news that R1B1 was split, and there's a new R-L754. I think I got that right. I'm not sure. RM343 was originally the designation for the R1B1 haplogroup. And you'll see it has 25,602 individuals in it. And then the next SNP that comes off of RM343 is RL754. And that one has 25,571 individuals in it. These are people who have tested at Family Tree DNA. Then we get down to this one here, which is RBY14355. Now, BY stands for Big Y, and it means that this test was done at 23andMe. At Family Tree DNA. And you'll see that there are 29 individuals in this group. Well, I believe this is the split. Originally, this was the only haplogroup off of RM. Now we've got 2 haplogroups off of RM, and this one has 29 individuals in it, and then there are even more differentiations down below. And the way this was discovered was, Here, let me move this there. They were doing testing in Central Asia, and they came up with a SNP mutation that they had never seen before. And they found one example of it in a Hazara ethnic person, one in a Kazakh, one in a Tajik, two in Turkmen, one in Uyghur, and one in Uzbek. Now, this is important because up until they found this, they thought that all of these people were in the same haplogroup, that they all matched, that they all had the same SNP mutations. And basically what they found was a mutation way back in the ancient DNA that they had not found before. And that caused a split in this R-L754, because these people here, these 25,000, do not have the same biological haplogroup as these 29. Now, my question is, by explaining all this, do you understand what happened here and why this is important? Because essentially it separated the R1B1 into two haplogroups. So does anybody have any questions?
Nothing in the chat box right now, Marleen.
Okay, Suzanne, are you satisfied with the explanation?
I mean, I really appreciate this because it actually kind of took your last presentation and made it a little clearer to me.
Yeah, well, I talked about the same thing in the last presentation because I was trying to show people the importance of the terminal SNP. And this is not necessary. Well, this could be a terminal SNP. Yeah, this would be, these would be terminal SNPs in the sense that their genome is so old that they haven't had any mutations after that. But I was trying to, in my last talk, I was trying to show the importance of terminal SNPs, because terminal SNPs tell you where your ancestors came from geographically. And my terminal SNP for my male ancestors is so far down the phylogenetic tree because the mutation that created the I haplogroup happened so recently in time. I mean, we're talking about, say, 2 or 3 million years of evolution here. And my people only evolved 2,000 to 4,000 years ago. So it's a drop in the bucket. But these people their genomes are old. You can probably identify these people, you know, by just testing the first 12 markers. And you wouldn't get down here because family tree DNA doesn't tell you this much information. That you have to do a big Y to get this information from family tree DNA, or find somebody, a couple of people who have had the big Y and who ended up with this SNP designation. Then you can go to Thomas Cron, and for $20, you can test yourself for it. The only problem with that is family tree won't let you allow it in, won't let you post it in the haplogroup designation. And on my brother, what I did was in the column where you indicate your earliest known ancestor, before I wrote my ancestor's name, I wrote the SNP designation that had been tested someplace else. But I recently, talking to the group support at Family Tree DNA, I was told that if I can get 5 people to go together, the SNP cost at Family Tree DNA, the SNP costs $39, $40. So if I get 5 people to go together, each contributing $40, that their SNP designer can probably go in and design a specific SNP to look for SK1234. And then they'll put it on the SNP list as something that can be purchased. And the other members of my Van Horne project who descend from Mathis Corneliuson can go in and get a SNP test that makes our terminal SNP all the same.
Marleen, DP put in the chat box, this has helped me understand how my family got down to 7 individuals from thousands on R-M269.
Yeah, but R-M269 is not the terminal SNP. Does it show up in red? If it shows up in red or green, RM269 is way up the phylogenetic tree. It's maybe 10 removed from the trunk. And this is maybe 100 or more removed from the trunk. It's well, maybe not that far because it's so old, but it's way further down.
Yeah, Arlene, we've actually done the WISE 700 testing, so we're way down.
Oh, and it's just RM269?
RM269, no, that's the top level, but no, now we're way down to
Yeah. And
like
if you
R-FT da.
yeah, okay, R-FT. The FT means that is a SNP that was identified at family tree.
Mhm.
And this BY on this one here means that this is a SNP that was also identified at family tree as a result of a big Y. Okay, that just to make sure that people understand that if they have a red SNP or even a green SNP that only has 3 numbers in it, that it is not your terminal SNP. It won't tell you where it comes from. It'll just point the way. It'll say go east, go west. But that's the most you're going to get out of one of those 3 digit identifiers. That's why I talked about this, because if you have a bunch of people that match you, or where a big Y has been done and you know what the terminal SNP is, then you can go to Thomas Cron at YSeek and ask him to test for it. Now, Thomas has a limited range that he can test for. He is primarily involved in I haplogroup. I have had others that I have asked if they could test for, and he's not set up to do it. Because he makes his own primers, and he's got thousands of them. He's been making them for 20 years. In fact, for a period of 5 years, he worked at Family Tree DNA in Houston, and he set up their advanced testing lab, which allows them to do this. And then his contract was probably up or whatever, and he and his wife moved back to Berlin. But the YSeek, if you do one of these tests at YSeek, mostly if you tell him what you're looking for, it's $20 is all it costs. And you don't have to do the big Y. Somebody has to do the big Y to get you on the right track. But once you're on the right track, you don't need to do the big Y for every single person that comes along. All you need to do is test them with Thomas and see if they're positive for that SNP.
Now, Marleen, how would somebody apply this to their tree? You know, what would this do for their tree?
Well, it depends on their tree. And quite frankly, this split here has no real significance for anybody but these 25,602 people. Why it was so newsworthy is that it's a SNP, a 17,000-year-old SNP, I think is what they said. It came into the genome 17,000 years ago. Well, that makes it interesting because back around that age, they thought they had found all the SNPs, you know, the geneticists. And they didn't realize that, well, here's a branch we didn't know about. And that's why it's important. It doesn't really affect anybody unless you're somebody who shows up with that SNP. And then it practically means that you probably had Hazara, Kazakh, Tajik, Turkoman, Uyghur, or Uzbek ancestry 17,000 years ago.
Okay, that answered the question.
One of the things they found when they started using SNP IDs and SNPs to identify haplogroups is they found that they had haplogroups that were misidentified. And now this wasn't misidentified in the sense that it was wrong. This was misidentified in the sense that they hadn't found the appropriate SNP to make that change. And this is going to happen all along. Remember, I said that my cousins and my brother are ISK 1234. Well, they're not anymore ISK 1234. ISK 1234 was what they were maybe 4 or 5 years ago. Now, more SNPs subsequent to ISK 1234 have been discovered. And some of the people in my Van Horne project who have done big Ys now have a SNP that is like down here. If you imagine that this here is ISK 1234, and then it has some other SNPs hanging off of it, like this does. Well, the SNP that actually identifies the most recent terminal SNP for my family is one that's down here, and it has a different name. And so this business, this SNP business is it's like a floating raft, or it's in motion. It's moving. The more recent things they find, and in this particular case, they found an ancient SNP, then it changes the tree because it adds a branch into the tree that wasn't there before. But unless that branch affects you personally, it doesn't mean anything. And that's pretty much the end of my prepared talk because I've run out of information.
Okay. Well, does the class have any questions? We'll give it a few moments, Marleen. If you do, unmute your microphone, but leave your camera off. And this slide that you have on the screen right now, this is all so far back in time. It's obviously prior to 500 years ago, correct?
No, it's 17,000 years ago.
Yeah. Yeah. So it's before genealogical research ability time.
Yeah, before genealogical time.
Yeah.
But you're going to find people today. I mean, obviously, these gentlemen down here on the bottom are living today, and they have that SNP. And they may have more subsequent SNPs, which actually, let's just say I don't know what the new SNP is called. This one with all these people in it are not these guys down here. I think these guys down here are in this group, but I could be mistaken. But you can see how we've got a split here between RM343 originally had this, and now they know it's got this too. And actually, somebody living today could be one of these people, but their path through the phylogenetic tree would go up here like this, and it would go over here like this to here. And then it would go up until it got to the trunk of the tree again. And there's a whole bunch of SNPs before this that would register.
Would it be fair to say that the ones with the lower numbers, like 25 and 2 and 29, that number will go up as testing continues, correct?
It should. It should. Now, another question is that have all these 25,571 people who are theoretically in this group, have they all been tested to see if they're still in this group or if they should move down here? Because this sample that was used to get this new SNP was only 157 people. And the R1B1 is a major haplogroup for Europeans. R1A1 is the largest haplogroup for men in Western Europe. And R1B1 is the second, but it's quite a bit smaller, if I'm not mistaken. Because like right here, we're only talking about 25,000 people, and the Y-DNA database set at Family Tree DNA, I think, is 1 million. But most of the men in Western Europe are R1A1, and then the next largest group, I believe, is R1B1. And those are people from continental Europe. Now, my ancestors come, the I haplogroup is pretty much located in the Scandinavian countries and Denmark. And that's where my male ancestors come from. And that's what I explained in my previous talk, where I was able to show you all the incidences of SK1234 that appear in southern Sweden and in northern Denmark. And to go back and to say this again, when DNA testing first became the thing, the National Geographic hired Spencer Wells to go all over the world and take DNA samples from anybody he could find who was willing. And they mapped those. And so that's how they know where these samples come from. And that's one of the important things of doing SNP testing, if you can do it without having to do the big Y, because it will tell you originally where your people came from in the world.
Which can be a very interesting part of your family story when you publish it.
Well, yes, and in the case of my family, my immigrant Van Horne ancestor came to North America in 1663. And when his sons chose a surname, let's say around 1714, they chose the surname Van Horne. Now, prior to that, there had been an immigrant from Horne, Holland in the 1650s. And so when my family chose the name Van Horne, the immediate assumption was that they were Dutch and they came from Horne, Holland, where this other gentleman came from. Well, I'm telling you, it's been 350 years that my cousins have all thought they were Dutch. And they're not Dutch, they're Danish. I have some cousins that are pretty annoyed with me. And that's why the origin of my immigrant ancestor, it hasn't been known until, let's say, 2020, that we had irrefutable proof that his haplogroup, by that I mean the haplogroup of his descendants, is centered around the northern tip of Denmark and southern Sweden. It appears in one other place. It appears in England, west and north of the town of York, the city of York. Well, what a lot of people don't realize is that in the year 900, the Danish sent a bunch of colonists and established the community of Jorvik, from which York is an evolution. And the Danes lived there for a good 200 years. Now, those English people who match the haplogroup of my ancestors are descendants of some of those settlers to Jorvik. That to speak about them, they want to be Dutch. I mean, they want to be Scots. And so they're sure that they got that haplogroup because their Scots ancestors were living on the coast, and when the Danes raided and raped the women, that's how they got it. But I'm almost certain that those people who have that haplogroup descend from the Danish settlers of York and Jorvik. And I think that's more exciting than being raped. I don't, that's a real historical thing. But after about 200 years, the Danes abandoned the York settlement, and you had people who had lived there for 200 years. They weren't really Danish anymore, and they just stayed, and they went out into the English population. And as I said, they moved west over to Lancashire because there are some people with this Danish haplogroup in Lancashire, and there are some north. But for the most part, they are settled around York. And in fact, the surname on those people is Wood.
Marleen, DB put into the chat box, Family Tree DNA has a chart on the Big Y test results page that shows how the matches break down. For example, it would show the 25 people that make up R-PH155 and the 2 people that make up R-FTB1. We used to see this how our we use this to see how our 18 people split through descendancy to 7 different families today.
Yeah. In fact, I got this off of Family Tree DNA, but when I looked for the explanation of what they had for the makeup of R-L754 and RBY14355, they didn't have any explanation. So, this is as close as I could get to the information that you wanted to see what happened here. And this information down at the bottom comes from the paper that you sent to us in the email. I don't know if you just sent it to me or if you sent it to everybody else, but you might include the URL for the paper announcing this so they can read it and they can look at the charts themselves.
I'll look back. I may have already sent that out at my Friday links. You used email to everybody, but I'll double check that.
Yeah, you might have. And if you have that, all I can say is for those of you who are listening to this, go look at it. It's kind of dense. I had to look at it several times to make sure I was getting it right. And in fact, I realized that I had the wrong snip here this morning, and I had to take this out and redo this morning. But it also, if there is a chart that's got little green boxes across the top, and if you click on it, it'll expand and it'll show you of the 157 people that they tested, how they broke down and what they broke down into. And it shows you this one here, which is new, or however it turns out to be new. This one that was split. And these are the ethnic origins of the people who tested for that new SNP. And
Okay.
so
I'm
my
sorry.
question is, does this really help you understand whatever Y-DNA testing you've already done, or why Y-DNA testing is important?
Okay, class, this is your chance to respond, either in the chat box or Let's see here. Okay, Marilyn, I'll answer that separately.
Hi, Marleen.
Yeah.
This is Hugh.
Hello,
Hi.
you.
I was going to get a hold of you earlier. I haven't. Can you look up my kit number and get it from there?
I can't do it here on the Open Lab, but if you want to contact me personally, then I can do an analysis for you and tell you that.
Okay, well, I have messages from my great grandfather that my last name came over from England. There was 5 brothers.
Okay, let's
they-
stop it. Let's stop that right there because this is being recorded and this is not part of what we're talking about here. That's
Okay.
something you need to talk to me about personally.
All right, I'll give you a call later, maybe tomorrow.
And Hugh, I put Marlene's email into the chat box for everyone to see.
Okay,
Oh, okay.
write me an email and I will make an appointment with you. I do this by appointment. It takes 2 hours for me to go through and explain either Y-DNA or autosomal DNA, depending upon which one we want to look at. And so I'll do both, but it'll take 2 hours for each.
All righty, thank you.
Okay.
Okay, Marlene, in the chat box, DB says, definitely help me understand it better.
Okay.
And Catherine
Good.
says, yes, it helps her understand better.
Okay. That's my objective. Y-DNA testing has been sort of overlooked when the other popular genealogy sites started offering autosomal DNA. Number one, autosomal DNA is very inexpensive to do. That's one thing. The other thing is when you do autosomal DNA testing, you get thousands and thousands and thousands of matches. Well, I have done autosomal testing at Family Tree DNA. I didn't do it anyplace else. And the reason I didn't do it anyplace else is that over 20 some odd years of being a genealogist, I became well aware that most of my Van Horne cousins have the wrong ancestors and the wrong pedigrees. Now, autosomal testing became available when I was 72 years old, and I did not want to spend the rest of my life telling my cousins why they had the wrong ancestors. So I never tested at any place else, and I never put any of my ancestors in GEDmatch. I have enough trouble just keeping up with the people who contact me directly. And I'm happy to help them. In fact, anybody on this, on Open Lab, if they want me to do a free analysis of their autosomal or DNA testing that's been done at Family Tree DNA, I will do that.
Okay, and once again, I did put your email into the chat box so everybody can grab that and email Marlene and set up your Y chromosome DNA test and get it analyzed for free. That's very generous. Thank you, Marlene.
Well, it's the least I can do. Besides, it's my social activity.
I think genealogists have a tendency to do that. They always associate with other people who are doing genealogy.
Yeah.
Okay, let's see here. Barbara says very generous, thank you. And so we're gonna open it up for just a few more moments before we stop the recording. And let's see if anybody has any final questions. And while I'm waiting for final questions to come in, either by unmuting your microphone or putting it into the chat box, I want to remind everybody that after Marlene's presentation is done, we do continue on with the second half of class. If you're new to the class today for the first time, the second half of class is where we all work together, either discussing genealogy topics or working on somebody's brick wall, whatever you'd like to do. The second half of class is where you want to take it. That's why we call it the lab, where you get to experiment and go in the directions you'd like to go. Let's see here. Randall says, I'm up on my Family Tree DNA. Okay, so Randall, we'll take that up as soon as we stop the recording. Any other final last minute questions for Marleen? Okay, Marleen, it looks like that's it for the questions for today, and we really thank you for your time and your expertise. And at this point in time, if you'd like to say goodbye, wait for you to do that before I stop the recording.
I'll say goodbye and thank you for being willing to listen to me.
And thank you, Marleen, and thank you for letting us record today as well. It's very much appreciated.
Hope it helps somebody.
I'm sure it will. Thank you very much, and I'll stop the recording.